and that is how many oceans are there, because it's a popular question at the moment.
We can get feedback on that.
Okay.
That's not a problem, definitely. So in the chat box, how many oceans? We'll give you a moment. Oh, by the way, Ian, Maria is a fellow Brit. She was very excited in the chat box there.
Oh, good.
That she had a kindred. Oh, there's no, come on. How many oceans?
I've actually led you astray with putting those numbers up there, because it's not one of those, but that's usually what we get.
You don't have anyone. Oh, one. We've got one. Maria said one. Simone, any comments from your group?
Come on, Simone, you know this one.
It's all connected.
Who said that?
Maria.
Oh, okay. Should we'll put everybody out of their misery? Absolutely. Good work, Maria. Excellent. It is all connected, isn't it? So really, at the end of the day, we are only looking at just the one ocean. So we need to remind ourselves that what we do at certain areas in our ocean actually has an impact elsewhere. The ocean. In UNESCO and through America and Europe at the moment, the key words ocean literacy are bandied around a tremendous amount. There's also the MISA over here in Australia also spoke about ocean literacy a long time ago. It's good to see that ocean literacy, although the terminology is not being used in curriculum, it's still at the forefront of education in Australia. The new syllabus for biology and marine science and also earth and environmental science, they leave great scope for looking at ocean literacy and putting it into the syllabus. So when we talk about ocean literacy, these are the 7 tenets that generally we speak about, and that's being the Earth is one big ocean, you know, with many features. I mean, let's face it, it's sort of it covers the entire globe. So the ocean and the life in the ocean, it shapes the features of our Earth, you know, right from the beginning of time, and still does so today with movement of sediments and erosion, you know, transportation, deposition all around the world. The ocean also makes our Earth habitable. I mean, the vast majority of oxygen that we're breathing is actually coming out of photosynthesizing organisms in our oceans. It also supports great diversity of life, and obviously there's a whole variety of ecosystems. Even if we just look at the climates that it covers, being the polar regions and down through temperate regions, subtropical, tropical, etc. And the humans, I mean, we're inextricably linked with the ocean. There are huge numbers of people. Oh, goodness me, I should have. There's something in the region of, it's in the region of about 500 million people that actually rely wholly and solely on the ocean. It's not because they like fish and chips. It's because that if they didn't have the ocean and the resources that it provides, they wouldn't be able to survive. So very significant part of our planet. And to be honest, for the last one that you can see there, it's largely unexplored. A tremendous amount of work is being done by lots of different organisations, particularly with regard to the deep abyssal trenches and benthic studies that are going on at the moment. So this is the sort of information that I believe we need to be sharing with our young people coming through, so they are truly ocean literate and can actually make decisions when they're put in a situation where they're being asked whether it's a good thing to do this or whether it's a bad thing. What do you think about it? We need our young people to actually be able to draw upon a knowledge and make a reasonable decision with regard to that. So just as an overview of basically what's happening with our changing oceans at the moment, obviously global climate change is having a massive impact. This is driving ocean acidification. So the large amounts of carbon dioxide in our atmosphere are being absorbed into the ocean waters, and they're producing carbonic acid. And when that dissociates, we're getting the hydrogen ions bonding with carbonate ions, and by this bonding taking place, it's basically removing those carbonate ions, which are very important for making the shells of the vast majority of invertebrates that we have living in our oceans. It's actually basically tying up those carbonate ions, and those are then not available for the organisms to use, and so they're compromised right from the get-go. Other issues around acidification, it also affects animals' behaviour. This then in turn affects biodiversity, distribution of species. There's a whole gamut of influences that are driven by global climate change that we really do need to address. Okay, so our Changing Ocean program that we've developed here at Jacobs Well, it's taken into consideration that there are certain parameters in our ocean which are going to be changing over a period of time. And so what we've done, in order to start that discussion, we've developed a program that fits into the Earth and Environmental Science, Biology and Marine Science syllabus. And it looks at ecosystems and their diversity, and it asks questions of the students. They need to gather data in order to be able to answer. So that we selected two areas that we would like the students to actually compare. So if I can, at the heart of the program is basically about collection of planktonic settling organisms. Now, when we talk about plankton, plankton is basically organisms that live in bodies of water that can't swim against currents, tides, etc., wind currents. They can only basically go wherever these currents take them. Most of these organisms are very, very small. They can be planktonic for a short period of their lives. The mirror plankton, for example, crabs, they float for plankton for a period of time until they get large enough and they've eaten a tremendous amount of other plankton, and as they grow and get heavier and they fall to the seabed, then they're out of the plankton then, and they're quite mobile though. They run around and they scavenge. The organisms that we're interested that are settling out of the plankton, they are sessile settling organisms. So they're organisms that people don't normally consider as necessarily being, I suppose, worthy of discussion. They're not charismatic marine organisms. They're not well thought of, to be honest. The organisms that we're looking at, most people would scrape them off of the bottom of their boats, and they think they're a bit of a nuisance. And we're talking about organisms like barnacles and tunicates and bryozoans, tube worms, etc. All of the stuff that people put protective paint on their bottom of the boat to stop gathering, we want it to gather on our equipment. Now, the equipment that we use was based upon the autonomous reef monitoring structures that were developed by NOAA, the National Oceanic and Atmospheric Administration in the United States of America. These devices were put together to be placed amongst reefs during the ocean census. So the ocean census was an initiative where we wanted to find as many organisms and identify as many organisms in the ocean as we possibly could. Now, the idea of a coral reef with its three-dimensional structures, there are a lot of little critters that live in between those staghorn corals and the big brain corals and the boulders. And this device was actually placed in situ for a 12-month period, and it was basically an invitation to come and settle there. So NOAA were using this to actually collect all of these organisms. They were then scraped off. That's the ones that were large enough were identified visually, but then some of the smaller elements that they couldn't necessarily identify just by staring down a microscope because it would take too long. They were identified by genetic markers. Now, here at Jacobs Well Environmental Education Centre, we don't have that sort of technology, and I think it would take longer than a 2-day camp to actually fulfil that. So what we did is we borrowed the idea of these autonomous reef monitoring structures, and we adapted it. We removed all the small homes that were found in the NOAA structures. But we also added these devices. So rather than the structure sitting alone on the seabed, we also attached some data loggers. Now, notoriously in studies in schools, if you go out and do an ecological study, we'll go out with a whirling hygrometer, and so we'll whirl it around our heads for a period of time, and we'll say at this point in time, this is the amount of water that's in the air, so this is the humidity, or we'll say this is the temperature at the moment. But these are all moments in time, and I think it's to come up with an investigation which is significant. I think what we needed to do was to actually collect data over a long period. So we attach these data loggers. So the green data logger you can see at the top there, that measures temperature and light, and the logger that you can see at the bottom, this is a current meter, is developed by James Cook University. Here's one I prepared earlier. So this one's come back. We've got to recalibrate this one, but that one basically measures water current, and it measures it by how much tilt. When the water current is flowing past it, it knocks it off of centre, and it acknowledges how far off centre it's gone, and then using the algorithm in the software, it tells you how fast the water's going. Fabulous bit of kit. We can record this information every. Well, at the moment, the water current meter is being recorded every 10 minutes, and the light and temperature is being recorded every 5. So this is one of the sites where we're currently looking at. This is off of Wavebreak Island in the mouth of the seaway. So this is our final arms unit with all the data loggers in place. Now, I noticed on that previous slide, I've actually said it was 10 weeks. We've changed that period of time that it's deployed for from 10 weeks down to 7. That's purely because of the amount of marine growth that we get on these plates over that period of time. So there's 2 places where they're found. This is a quick video of, oops, maybe not, we won't worry about that video. I do apologise. There we go. It's not very exciting anyway. So this is one that we only pulled out it was earlier this week. We pulled this one out earlier this week. You can see the amount of growth on it. There's a few down there. We do have a tremendous amount of trouble with algae and algal growth, particularly when it covers the light data loggers. So it involves us going for a bit of a dip every couple of weeks just to clean all the data loggers. So it's quite labour intensive. Now, we need to, once the loggers have been down for a period of time, and they've been down for that 7 weeks, what we need to do is we need to start identifying some of the organisms that are actually on there. Now, as I said, these are not charismatic animals. We're not looking at dolphins and whales and sharks and those larger animals that when you're young students, they get really excited about them, and I mean that's one of the reasons I'm where I am, is because of that splitting movie Jaws. I mean, at the time I wasn't actually allowed to watch it. I was a little bit too young, believe it or not, but I did read the book, and I found out about this thing called marine biology, and I thought, oh, I like the idea of that. So anyway, here we are today. These are not charismatic sharks. These are organisms like barnacles, bivalve mollusks, the jingle clams. We've got encrusting bryozoans, tunicates, tube worms. So we've also got some algae on there. I'll get into that a little bit later on. But these animals are significant parts of our marine environment. They're pioneering species that inhabit a newly available area. They change obviously the biota of that structure that's underwater, and by making themselves available as a resource of food, it attracts other organisms to that area, and so you increase the biodiversity purely from the settlement of these organisms. So these organisms are very important, and that's just one reason why they're so important. So some of the organisms we can see here. So this is the collection of the biotic data. The first ones that the most prevalent that we see are the red striped barnacles. We find tremendous numbers of those. They settle out of the plankton, and they find an area that's a hard substrate. They also use their antennae actually to look for evidence of other barnacles having settled in the same area. And if they do find that evidence of the glue, they'll put their head down, bum up, and they'll settle and build their calcifying shell around them. Now, these guys are filter feeders. Most of the time they'll draw in anything that's available, but phytoplankton would be one of their major food sources. And phytoplankton is the plant plankton, as opposed to the zooplankton that is also available in the ocean water. We also have these organisms, these individual tunicates, or the common name is sea squirts. You may have trodden on a few of these if you're scrambling over the rocks in the intertidal zone. These organisms are when they settle, prior to settlement, when they're actually in the planktonic form, they actually have something called a notochord. So it's like a precursor to a backbone. So that is not actually, it doesn't develop further when they settle, and so they are not vertebrates, but they are invertebrates. You can see from this structure here, this is tunicate anatomy. They have an inhalant siphon and an exhalant, and we can. Oh, can I point to? Can you see my cursor? Can you guys see my cursor moving? Oh, good.
We can see it.
Okay, so you can see our intake here. So what they basically do is draw water in through this siphon and exit it through this siphon. Now this area here in the pharynx, it basically has the ability to secrete a really fine it's a net. It's basically a mucus net, and if you were to see micrographs of it, that would have been a good idea to put one in. The micrograph of the net is very, very fine, and that's where a lot of organisms will actually stick to. So this is how it feeds. So this is its gut, and you can see it now feed down into the intestine. You'll also see that it passes out here through the anus, so all the excretory matter comes out through the anus, but also you can see here the genital duct that's next to it. So this organism actually has ovaries and testes, so it's a hermaphrodite, is both sexes, but they want to try and avoid self-fertilization, obviously, so they can ensure that there's diversity amongst through their species. So what they'll do is they'll release the sperm and they'll release the eggs at different times, so within different seasons, and that way they try and avoid self-fertilization. So they're very, very interesting organism. Here's some tunicates they're colonizing. These are not individuals. They're the same organism though, but these organisms work in colonies. Now this one here was taken from our microscope recently, so you can see the opening here that's the inhalant siphon, and now you'll notice as it sort of fines off down to the bottom here that the exhalant siphon would be down here, and this area through the middle there, it's basically where all the excretory materials will come out from for this group of colonizing tunicates. So it's kind of like it's like a shared bottom really. So those are our colonizing tunicates. Here's an example of this is I think this is butcherless species of colony of tunicates, and this is them not settled on one of our arms unit. This is it settled on some holophila in the field, and there's quite large quantities of it down there. We also get encrusting bryozoans. These bryozoans produce a skeleton or a structure that's made from calcium carbonate, and each cell, can you make out the individual boxes here? Each one of those individual boxes will contain a zooid. Now, these bryozoans is a little bit of anatomy for this one. I'm going to move along quite quickly, sorry. Again, they have both ovary and testes, so they're hermaphroditic. They will release their gametes at different times, again, to avoid sort of self-fertilization. These guys, can you see these long structures here that make up the lophophores? Okay, it's like a flower. In fact, when you see it under a microscope, it's very reminiscent of petals on a flower. But down each one of these strands, they have cilia which beat quite rapidly, and it basically produces a current. So the current will draw in organisms such as phytoplankton that they will feed on, and that will also enable them to get the oxygen that they need. All right. There's another example of a bryozoan. These ones don't exist in the flat colonies. These are the branching colonies. Now these guys are they get mistaken for algae a tremendous amount, but they are actually animals. So again, there's a whole colony of animals, and if you can make out the small little cups down the side of each one of these branches, they all contain the individual bryozoan. Now, our data collection. So what we're interested in finding what, can you guys see me? It's getting very dark here.
Yes, we can. We can see you. Right. Yep.
So this data collection sheet, we want to identify the organisms that are there. We want to identify how many of them are there. And in some situations where possible, we'd also like to identify the size of those organisms. The areas where we're actually placing our arms units, can you see this large map on this side? So this is the entrance for those people not on the Gold Coast. This is the ocean side. Okay, this is South Stradbroke Island, and we have an inlet, the seaway, where the ocean water comes directly in, so it can go behind South Stradbroke Island. This is Wavebreak Island, and we have one arms unit, or a number, depending on the time of year, that are placed at Wavebreak Island. So they're exposed to really good quality ocean water, carrying all those planktonic organisms. Now that water makes its way up through the Broadwater system on the inside of on the inside of South Stradbroke Island. This area here is there used to be an environmental ecological resort. It's Cove. It has a marina, and we also deposit an arms unit in there. So they are very different physically, but they're carrying in their water the same planktonic organisms. So these organisms still have the capacity to settle on the arms units, either at Wavebreak Island or at Cove. Now, dependent on the water, depending on, in this case, we're looking only at the physical factors, dependent on the physical parameters, that sort of denotes whether the settling plankton are successful or not. So if I can just show you, this picture was taken at Wavebreak Island in the seagrass meadow. So each unit is placed in, obviously, between 4 to 5 meters of water because of the tidal fluctuation. So that's a view of Wavebreak Island. So that's what the arms unit is exposed to there. Now, our next view is of Cove Marina, which is, technically speaking, the same body of water. That's the view from the bottom of Cove Marina. So yeah, certainly not as pretty. Now, what we do with our arms units when we evaluate, we're looking for, we're counting organisms, we're measuring them. We compare spatially, so the difference between the two sites. So we will compare Wavebreak Island for a 7-week period compared to Cove Marina for a 7-week period. So they were exposed for the same time at the same time, and we will identify the organisms. Now, just by looking at this plate, I'm not going to go into massive detail, but just by looking at this plate, you can see the standout organisms here. Can you see those coppery, metallic-looking shells there? So those are our jingle clams. Awesome name. We've got a large number of jingle clams on the underside of plate 2 in February 2020. That's at Wavebreak Island. Yet at Cove Marina, on the underside of exactly the same plate number in exactly the same time frame, we only have 1. So, and if we look at the size of that in relation to the size of those other organisms, I think these organisms tend to be doing a lot better. Now, what we do with that data then, we will statistically analyse it to make sure that what we're looking at here is significant data. So what we do is we run a t-test on, in this case, it's the abundance. I'm sorry, just checking. Yes, so this is the mean abundance of jingle clams between the two sites. Now, each one of those arms units has 6 plates to it, so we analyse both top side and bottom side. So the numbers are quite significant when they come in. This is our mean size at Wavebreak Island and our mean size at Cove. Just by looking at this graphical representation and looking at the error bars also, and the fact that they don't overlap tells us that this must be significant. And when we look at our p-value, so we can see it's a lot less than 0.05, and hence it's a significant difference between those two sites. We then we can also, for example, with the jingle clams, because we've measured the size of them, we can also look for whether the size of jingle clams the difference in size of jingle clams between the two sites is significantly different also. And one of those, is this one? Where are we? Yes, jingle clam size. So when we compare them in our t-test, we have a 0.0, yes, a lot less than 0.05, and hence the data we have here, it could be suggested that the size of the jingle clams between the two sites, Cove, grows a lot smaller. We can also apply our statistical analysis to our abiotic data. So in this case, we're looking at water current speed. So this is the water current speed between Cove and Wavebreak Island. And our t-test tells us, this one I got a little bit scared about. I had to go and speak to a statistician about it. This analysis was performed on Excel, but because the value is so small, I thought this was a mistake. But it's because the value is so infinitesimally small, because of the amount of data that was collected. This is indicating that it is a truly significant value. So even though it just says 0, not a 0.0 something, it's certainly still a significant difference. So we can compare our water current means. We can compare our the amount of light. So this is an indication of the amount of lights that's hitting those arms units. You can see there a large difference again comes in at 0. Water temperature is very different between the two sites at the same period of time, and that's quite important to note. In the fact that the water, if we look at that at Wavebreak Island, the water that's coming in here at this particular period of time, we're looking at sort of 21.7. Okay, whereas at Cove, the average was 19.7. So that's quite a significant difference. Now, so we have obviously this body of water and an inflowing tide it's a great heat sink, and it can maintain a reasonable temperature over a large distance because of its depth. Whereas when we look at Cove, once the seawater has entered that broadwater system, we're only looking at an average depth of around 4 meters anyway. And so, of course, the exposure to increased air temperature, etc., can be significant and heat the water up. Or alternatively, in this case, the exposure to colder temperatures over winter can also reduce the temperature of the water. Okay. So what we ask our students to do with this is we're asking them to evaluate two sites in one period of time. Now, what we considered after that was, well, if we're going to be collecting all of this data, why can't we make the data available for other interested parties that could compare the same sites but over over a period of time, as opposed to spatial comparisons, where we were thinking of looking at temporal comparisons. So we developed the International Marine Science Database. The international part is because what we want to do, we've had information from overseas, the Marine Biological Association of the UK and the European Marine Science Educators Association, and also the Ocean Conservation Trust are interested in getting this program over in Europe, and hopefully we can then get spatial changes. We can get all this information from a lot of places around the world. At the beginning of next year, we're starting the first monitoring up on the Great Barrier Reef off of Lady Elliot Island. And so this International Marine Science Database is a place for students to go to obtain all of this information so they can use them in their own reports. Oh, goodness me. Cove, Marina. This is some of the temporal data. So what we're looking at here is the same plate. Okay, so it's the underside of plate number 1, but this one was in October last year, this one is February this year, and this one is July this year. You can see roughly most of the organisms on here are all bryozoans. Most of the organisms on here are tube worms with some bryozoans. And then when we look at this one, around the edge, we have some bryozoans, but all of these dots through here, it's difficult to see here at the moment, they're all tunicates. So we've not actually recorded a number of tunicates. This is a large tunicate, an individual. We've not actually recorded that sort of number in Queensland Cove Marina before. Somebody actually made a point. This is something that would need to be obviously investigated, but Queensland Cove Marina at this present point in time doesn't have a huge amount of boat traffic because the resort itself is shut. So during this time of COVID-19, the water clarity would appear to be improving. I think it's also helped by the fact we've not had a tremendous amount of rain, but what we did notice on the data logger that was recording light was a tremendous, was an increase. It was actually registering. So a lot of light was actually getting down to these organisms. Now, the turbidity or the amount of particulate matter in the water at Queensland Cove Marina is usually quite high. That reduces the light, but it can also, if it's a sediment, impact on the feeding of a lot of these organisms, which are indeed filter feeders. So that's something that we're going to look into further. And it's interesting to see as this program has developed, we're actually coming up with our own questions that need answering. So I do think eventually what we're going to do is turn this into some kind of investigation. Here's some plates from Wavebreak Island. Again, this is over time. Down in the bottom left-hand corner here, you can see October 2019. These organisms here, these dark brown blobs, these are all tunicates. We've got a couple of jingle clams in there. But then in February, so this is springtime, coming into spring. This is at the end of the summer. Look at the number of jingle clams. It's phenomenal numbers that we had. And then in July, so in winter, a couple of jingle clams. We also have some tunicates, but not very many. So you can see how these plates, they do change over time. Now we're going to build a whole database with this information. We're going to look for change over time. We're also going to be able then to look at the abiotic factors, so the light and the water temperature and the water current, and those are the physical parameters that we're currently investigating. But with regard to the future of this, we're also we're in discussion at the moment. It's going to be quite expensive. What we're doing is we're putting together a buoy system which is going to measure pH, light, turbidity, and we're also going to be looking at the amount of phytoplankton that's in the water. So light, turbidity, pH, salinity, and what was the other one? Have I said water current? Water current's the one I'm after. So those 5 parameters. So we're actually going to start looking at the chemical difference in over time of these where these plates are situated. Now this information will be pinged either via the phone network. I'm not sure about how that's going to work on the Great Barrier Reef, or it's going to go via satellite. It's just that one's very expensive. So this information will be put up onto the website so we can make this abiotic data available for all schools that are participating in this program. And as I say, it's developing as we go. It's giving us more information about what's happening in our oceans and how the physical and chemical parameters are actually impacting the growth of these pioneering species. Because as we know, these pioneering species will then impact biodiversity actually down the track. So when we start thinking about our oceans and how they're changing, this is certainly not a political statement or a political arena. I'm just talking about we are seeing change. That's the fact. Change is happening. We know why the change is happening. It would appear that we're not going to see an abatement to the emissions of fossil fuels within the very near future. So I think it's important that we understand the changes that are happening in our ocean and we start to plan for management of it. So prediction of the future, what the future is going to look like in our ocean, is important so that we know how to manage it. So we need to take all these pieces of a jigsaw. So I know we've been looking at small pioneer species, but those small pioneer species have an impact on the rest of the biodiversity. And I think we need to get every piece of the jigsaw together so that we can actually start making that plan and strategizing for the future. Probably the most important thing, sorry, one last bit, probably one of the most important aspects of this is communication. I think we do need to take this information, turn it into something that's very attractive and easily read and easily understood, and get it out there into the streets so that everybody can become ocean literate. So going back to the beginning, our students at school need to be ocean literate. We need to ensure the public becomes ocean literate, and in that case, education is certainly the key to the success of and a cleaner, better future for our ocean. Whew. Only 10 minutes over. That was pretty good.
Thank you so much, Ian. I don't think anybody minded you going over at all. I love the whole concept, and I mean, maybe it's because I'm not a marine biologist. It's the first time I've ever heard the concept of ocean literacy, and I found that so intriguing. I've got so many notes down to look up later on, I have to tell you.